This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 65, recorded on September 26th, 2013. Hi everyone, I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, is Michele Swanson.
Hello, Vincent.
How are you?
Great. Beautiful weather here. We've had just warm, sunny days and cool nights.
It is getting fall-like, though, isn't it?
Yeah, it's great. It's my favorite time of year.
I don't know. I have to say I like the summer. I like the hot, sweltery, humid weather. I don't know why. The fall's okay, but I do like it hot, but it's no longer hot here. I just did a podcast with your colleague, Kathy Spindler, so I know the weather in Ann Arbor today very well. Here it's pretty much the same. It's about 68, 20 C, and low humidity, cloudy sky. It's really beautiful. It's a nice fall day.
Oh, I love it. Yeah. There's something rejuvenating about the beginning of a new school year. I guess it dates back to childhood.
I was talking to Kathy about that, how many students invade Ann Arbor in the fall, right?
Yeah.
It must be such a change in the whole character of the town, right?
It's quite different. And then because we have a lot of academics here who can leave for a month in August, even the neighborhoods get quiet. So,
gosh, that's 40,000 students, right?
Yeah.
It's incredible. Well, today we had hoped that Elio would join us, and he's not online. I hope everything is okay, but maybe he'll join us halfway through. We had two papers lined up to do, one by Elio and one by myself. So if you're game, Michele, we'll just go ahead and start on the one paper.
Let's do it.
This is a paper that is something I would characterize it as a spectacular result. It just came out in Science September 6th, and the title is Gut Microbiota from Twins Discordant for Obesity Modulate Metabolism in Mice. There's a whole slew of authors. The first author is Vanessa Ridara, and the last author is Jeffrey Gordon. I know you tried to contact Vanessa to get a little history of her.
Yeah, she is busy. She is going to defend her thesis in just 2 weeks. So she is assembling all her research into I'm sure, what will be a beautiful thesis.
Yeah.
I can tell you that she's from Venezuela and did her undergrad degree there, was a summa cum laude graduate, and then started graduate school at WashU in St. Louis, where she did this work with Jeff Gordon and their many colleagues.
Yeah, a lot of authors on this paper. As you'll see, it's a multidisciplinary paper, which is the way science is going. And I would never have known that she's from Venezuela by looking at her name there. It's great to be able to just get that one little factoid. Is really good. So this has to do with obesity and the microbiota. And we've had a couple of papers on TWiM where we talk about that topic, mainly from the perspective of comparing lean and obese twins usually, because that's a good pair to look at. So you control for genetic differences and seeing what the gut microbiome is. But as they point out in this paper, it's really hard to make conclusions from these kinds of studies because they're descriptive. You can't establish any causality. You can't say this population of bacteria is responsible for obesity or not. And that's where this paper comes in. They also work with twins. They use what are called discordant twins. That is, one individual is lean and the other is obese. So they have a lot in common, obviously, except for that phenotype. And then they harvest feces from them and administer them to mice. So they collected 4 female twins, and they're discordant for obesity. And they get fecal samples, and then they homogenize these samples and introduce it into mice. These are laboratory mice, They're germ-free C57 black mice, and they put a tube into their stomach and basically introduce the fecal suspension into them. And the idea being that, can you transmit obesity or leanness by the fecal microbiome? So they put the mice back in the cage and they feed them. They give them all the same kind of chow. They have a chow that is low in fat and high in plant polysaccharides. And in fact, in this paper, we're going to look at different kinds of chow, mouse chow. I think it's funny in a paper to read chow, but
yeah, it's what it's called.
That's right? What we call it, right? And then they characterize these mice. So there are so many experiments in this paper. We can't go into the details because there's just too much. I'm going to try and summarize them. But it's just a stunning result, and it's an incredible amount of work here. And I would really hoping that you would be able to talk to Vanessa, because I really wanted to know what she did. Because there's so many other authors on this paper, but I guess I'm not going to be
biochemists, probably the animal people.
Yeah. There's computer analysis of the microbiome data, because it's all sequence data that has to be massaged. There's a lot of statistics done. And so maybe Vanessa, well, who knows? I'd be guessing. I don't know what she did. So, all right, they take these mice and then they do basically sequencing of 16S bacterial ribosomal RNA. This is initially from the donor fecal specimen. So you want to know what these discordant twins had in terms of their microbiome. So remember that 16S ribosomal RNA sequencing can be used to identify the bacteria. And then they do the same with the mice, and they have time points. After feeding the mice the human fecal extract, they take samples of their fecal pellets and they sequence them as well. And the first stunning result is that the mice, the transplant recipients, they call them, they efficiently and reproducibly captured the taxonomic features of the donor human microbiota. So that means, the human gut microbiome is a very diverse collection of different taxons or taxonomic units, as it's called. And when you feed mice their human feces, that the mice end up having a very similar microbiome as the humans. And then, in addition to that, if you look at the proteins encoded by the bacteria that are transferred, you find that the mice have acquired many of the enzymatic functions from the donor. That's why they say the functions, they also adopt the functions encoded by the donor microbiome. So not only the bacteria, but their functions as well. But maybe the more stunning result is that when you take the microbiome from the obese twin and put it into mice, those mice get fat. And then then you take it from the lean twin and the mice remain normal weight. They use what they call quantitative magnetic resonance to assess the body composition. I guess it's a kind of nuclear magnetic resonance that can measure body mass. So you not only transmit the microbiome, but you transmit the body mass index of the twin by just giving them the bacteria. So why is that? So they look at the genes encoded by the transferred bacteria. They do RNA sequencing so that you can look at what these bacteria are producing in the human donors and in the mice. And what they can see is that the obese twins and the mice that get their microbiome have a lot of genes involved in detoxification, stress responses, certain kinds of biosynthesis, metabolism of amino acids, lots of metabolic activities that can be nailed down, basically. So the obese humans have a certain signature, if you will, of protein families, and that gets transferred into the obese mice as well. Some of the things that are notable is that the lean microbiome, if you will, has a lot of genes involved in digestion of plant polysaccharides, fermentation to certain kinds of acids like fatty acid, like butyrate, and other kinds of compounds like propionate and so forth, which are all have been associated with obesity. So basically you transplant the bacteria from a obese twin into a mouse, and that mouse can get fat. And looking at the genes that are expressed, you conclude that the microbiota from the lean twin has a greater capacity to break down and ferment polysaccharides than the microbiota from the obese twin and the obese mouse. So this is a lot of work so far. They're not only doing 16S sequencing, they're sequencing the RNA produced to look at what genes are made, and there's going to be even more. So that's why one of the reasons why there's so many authors.
Right.
So the next step, I suppose, is to know what bacteria do this. So they make a collection of anaerobes that they can actually culture. And there's a figure here that shows the list of them. Looks like there's about 40 of them, all different species. So they can culture these out of each of the twins.
Or we should say the ones they can culture.
The ones they can culture, because most of them can't be, right?
Right.
And then they feed these to the mice and see what happens. And they basically get the same result. If you take the cultured bacteria from the obese twin and feed it to mice, they get obese. Whereas the cultured bacteria from the lean mice, from the lean twin, the mice don't get obese. Okay, so this is cool because that means you can culture some and transfer the obese phenotype. So this suggests that, the idea here, of course, down the line is to be able to treat people for obesity. So if you can culture some of the responsible bacteria, that's good, I suppose.
A probiotic.
Exactly. I also learned a new word from this paper, prebiotic. Did you know that one?
And then they came up with yet a third one, the symbiotic, but we can get to that later.
Yeah. Probiotics is the one I know. Okay. Yeah. Now, the next series of experiments are even more spectacular. They co-house these two kinds of mice. So we have mice that have gotten a fecal transplant, we'll call it, from the obese twin and one from the lean twin. And they co-house them. And you might think, well, what's the big deal? Well, mice, in fact, eat their feces. They're coprophagic.
And their other neighbors' feces as well. Yeah,
not just their own, right?
Yeah.
So this is an experiment to see if you can eat your litter mate's feces and transmit the obesity phenotype. So they co-house them, and then they let for a while, and then they examine them. And the neat thing is that the lean phenotype can go from a lean mouse to a obese mouse, but not vice versa. So in this cage, the obese mouse gets leaner when it's co-housed with a lean mouse. Isn't that amazing?
I thought it was beautiful and obviously very hopeful. Because you might think, walking around our state fairs, that the obesity would be dominant.
Yeah.
But in fact, it was the lean phenotype that was dominant. Although, as you'll explain, it also depended on diet.
Right. So that's good in a way because, as Michele just said, you don't want an epidemic of infectious obesity. So it goes the other way. And you might think, wow, that's the right result that you would want. And in fact, That's what they got. They did a number of studies to basically show that a lot of the enzymes and pathways that were transferred were similar to the ones in the other experiment. And what they think is that basically the obese mice are lacking these members of this microbial community in their gut, and therefore they can be invaded by the community from the lean mice, but not vice versa. Because if the communities were the same, you wouldn't expect that to happen. But they call it there's an ecological niche in the obese mice, which is ready for the members of the lean mouse intestine to go in. And they looked at which bacteria were the good invaders, so they can do that quite readily. So the invaders from the lean mice to the obese mice, which then make the obese mice get skinny, they're members of the Bacteroides of all different genera. And they list them all here. And again, these Bacteroides go from the lean mice to the obese mice, and that's because they're missing in the intestines of the obese mice, so they have an opportunity to go in there and colonize them. And that's just by eating the feces again.
Just by eating the feces.
Who would know? I wonder
it's been right there under our eyes all this time.
That's what we had to do. Of course, humans are not coprophagic.
Under our nose, I guess. Under our nose.
Although dogs are coprophagic, right?
That's right.
Although most people who own dogs try and discourage their dogs from doing that. I think dogs will eat their own feces as well, as far as I remember. So unoccupied niches. Okay. Moving on here. So then they also did studies of the transcriptome, that is all the RNAs that are made in these bacteria, to try and understand what's the metabolic event that's going on. How do these bacteria help keep a mouse lean, these bacteroidetes? And what they find, and this is another set of experiments where basically they are taking samples from these animals and doing, I think, mass spec to identify different metabolites in them.
Right.
Huge amounts.
A real tour de force.
Amazing. It's a combination of everything. And basically what they find is that when the bacteroidetes come in the gut, those mice get better at degrading branched chain amino acids. Okay? And for those of you who remember your biochemistry, that includes valine and leucine and isoleucine, right? Amino acids with branched chains. And the presence of these has been correlated in people with obesity. Okay. And so here it kind of makes sense, right? These mice are better at degrading them. And so maybe that's one of the reasons why they are not obese. Branched chain amino acids. So I Googled branched chain amino acids, Michele, and I found already a health website that's selling them in pills.
Really?
To help. Because apparently if you eat the branched chain amino acids, it's good. But if you have high levels in your blood, it's not good.
So you need the microbes to metabolize them in your gut, it sounds like.
That's right. It's exactly it. And these bacteroidetes are the ones apparently that's doing it. There are also other metabolic studies that they've done that implicate other compounds, bile acids in being important targets for regulation. So bile acids are good to have. And the reasons why are rather complicated, but they're good to prevent you from getting obese. And they're also acetate, propionate, and butyrate, various acids that are metabolized by these bacteria. So basically, these are short-chain fatty acids, by the way, and these have been shown before to have a role in energy balance. So you can see that I think that the pattern that's emerging here is that, you have to have certain bacteria in your gut to help metabolize certain things that will be used for energy or other purposes, so that preventing obesity. Next, they wanted to see if they could culture bacteria from the intestines of the lean and obese twins, transplant those into mice, and see if they got the same effect. So basically, they took feces from each of the twins, the lean and the obese twin. They cultured them under anaerobic conditions and introduced them separately into germ-free mice. And what they found is that, once again, these are collections of bacteria, and they could identify what was in them, just the ones that would grow up under anaerobic conditions. And they found that when transplanted into mice, these cultures, these mass cultures or diverse cultures, if you will, stabilized in the mice. So they were able to transplant into the mice and stabilize and grow there. And like the uncultured communities, which were introduced into mice, these cultured anaerobic communities could also confer for example, an increase in adiposity of the mice when the mice were given cultured communities from the obese twin. So you take the cultured bacteria from the obese twin, you put them into mice. Those mice get obese. You take the cultured bacteria from the lean twin, you put them into mice. Those mice remain lean. So obviously it would be great if you could identify which bacterial species could protect mice against obesity. And unfortunately, they were not able to do that because in the next experiment, they cultured, again under anaerobic conditions, 39 specific taxa. These are called operational taxonomic units. They're bacteria defined by sequencing. They took 39 of those. Now, the experiment I just told you about, they took all the culturable anaerobes from the feces of the twins. In this case, they did just 39 of them. And they took those 39 culturable bacteria, or taxonomic units, and introduced those into germ-free mice. All right. So these are from the lean twin only. So these are lean 39 mice. They're called because they are lean from the lean twin, and they're 39 different sequenced taxa. These 39 cultured bacteria colonize the germ-free mice. They then put them in a cage with obese mice and ask, can those 39 taxa colonize the obese mice and prevent them from getting obesity? And the answer is no. These lean 39 animals, when co-hosed with obese mice, that is, mice that had received a fecal transplant from the obese twin, the lean 39 co-housed animals were not able to protect the other mice against obesity. Some of the 39 taxonomic units were able to invade the obese mice from the lean mice that they were co-housed with, and there were some metabolic changes detected as a consequence, but they could not protect the obese mice from obesity. So in other words, these 39 are not the whole story, and probably they're part of the story, but they have to be present in the context of a greater community. So clearly more work is needed to figure out exactly which culturable bacteria can protect against obesity. The effects of this are diet specific. They have two different diets that they use here. One has a low-fat, high-fiber diet, and the other is a high-fat, low-fiber diet. And they look at these 39 strains in terms of being able to transmit this phenotype. And what they find is that these 39 strains, when you get them from lean mice, they can ameliorate adiposity in other mice when they were fed a low-fat, high-fiber diet, but not if they are fed a high-fat, low-fiber diet. So, in other words, the microbiome is important in regulating obesity, but the diet is also a component of it as well.
Right.
You can't just assume that you can eat certain bacteria and that'll make you thin.
Yeah, it needs to be the combination, and their experiments showed that beautifully. It's really very elegant.
Yeah. Okay, so the diet and the microbiota are going to have to be really tinkered with in order to make this to work. So that's more or less an overview of this paper. Did I miss anything that's important?
No, I think that's really the highlight, that they have shown that we now have the methods to go in and really define a very complex population, measure the impact not only on the tissue but also on the metabolites, and then begin to make models that can then be tested in further experiments to really identify, hopefully, particular metabolic pathways or species that can be used to develop a treatment.
So basically, you want to know which culturable bacteria you could give people to help them maintain the proper body weight in the context of the right diet, right?
Right, which would be not only more appetizing, but also safer.
That's right.
Because the complexity of fecal material just raises the risk that you might inadvertently ingest a virus or a bacterium that is not good for you. So,
yeah, right. So currently people have been doing fecal transplants, and there has to be something better, right?
Right.
We know from giving blood, right? That alone can be dangerous unless you check for everything. And can you imagine having to check feces if you're going to transplant them for every possible?
Yeah, I don't see it happening.
It's not going to happen.
Not with FDA approval, but it may happen on the sidelines.
Now, there's a accompanying commentary in the same issue of Science called Fighting Obesity with Bacteria, and they make the interesting point that, so here in this paper, there's evidence that these Bacteroides species are protecting against obesity in mice, but they say that in human studies, the evidence is mixed, and there's a little bit of confusion in the literature. Sometimes they're associated with being overweight, and sometimes they're associated with not being overweight in people. And they say in lean African individuals, who consume diets high in fiber, they are reduced, these Bacteroides species in the gut.
Right. So I think although we'd love to know that it was a particular species and we only had to take that, it's probably much more complicated and not the species per se, but a particular metabolic pathway that could be provided by one of many different species.
Yeah.
Nevertheless, I think this is a great start toward defining what metabolic pathways we want to have active in our gut.
So the key here is that you have this nice mouse model where you can do this, right?
Right.
But the problem also is that it's a mouse and not a human.
True.
And so you have to validate whatever you find eventually in people. But you can do a lot of work initially in mice. You can dissect exactly what culturable species you need and then do the study. So this is really a beautiful model, and I think they're going to go quite far with it.
And it's encouraging that they can essentially humanize the microbiota of these mice to do these experiments. It's very hopeful.
I find that amazing. Right. But I guess knowing from the fecal transplant effects in people that this happens when you take up bacteria, they colonize you.
Right.
I know I've heard talks where people have monitored fecal bacteria, and as soon as you eat something different, your populations change. So we're very susceptible to it changing depending on what we ingest.
Which is good news.
Yeah, for this,
if we can be more disciplined about our diets.
But I mean, would you think that it would be so malleable, or would you think that it's really a fixed thing that's very hard to change?
I would imagine it's not going to change easily, but I would hope that. I know I made a joke about a colonoscopy in the past.
Yeah.
I would imagine there are measures that could be taken to help things, help the shift.
But we should point out that we're not talking about total shifts in the microbiome. We're talking about, the addition of these taxonomic units, which may not be present, or other kinds of shifts. But I think the core microbiome is more or less within a few different classes, if I remember some of those other papers. But it's cool that it can shift. And I suppose with something like the intestinal tract, where you're always ingesting materials on a daily basis, I guess it's not surprising.
And this will extend, studies are indicating this will extend beyond obesity and leanness.
Sure.
There's also some promising work being done related to colon cancer.
Right.
Actually, a group here in my department, led by Pat Schloss, has a paper that's being revised for our favorite journal, MBio, looking at the impact of the microbiome on susceptibility to colon cancer. Mm-hmm. And they also use similar transplant methodologies to show that the microbiome can really be protective, or a different composition can increase your susceptibility to colon cancer.
And this is in a mouse model?
That's right. Yeah, very similar principles they're applying. So, microbiology.
So guess
it's coming near you.
I mean, the simple explanation is that the bacteria are making metabolites that somehow are involved in
that can
cancer.
Trigger inflammation.
But you can also imagine that, your microbiome in the early life helps to mature your immune system, right?
Right.
So that could be part of it too.
And also, some bacteria make molecules that are that dampen the immune response. So it's probably some combination.
So this is revolutionary, basically, the microbiome.
I think so. And we have to emphasize it's because we now have methods to go in and catalog these very complex communities and do the companion biochemistry that we're going to be able to begin to tease apart this very complex biology.
So Carl Woese was a big starter in that, right?
Yeah, I also want to say this paper not only fascinated us, but the press has really jumped on it. CBS, NBC, the New York Times, the Detroit News have all carried this story, which is, I think, great for microbiology.
It is, yeah. Well, I think the microbiome in disease states has really catapulted microbiology into the news more than it had been for a long time.
Right.
It's a good time for Joe Handelsman to be in the White House.
It sure is.
All right, since Elio is not here, let's do a couple of emails.
Okay.
Maybe you can help me with these because I don't really know the answer to some of them. The first one is from Alexandra, who writes, Dear TWiMers, when I began listening to TWiM, to TWiV almost a year ago, I had just switched majors from philosophy to biology. I'm now writing to you good people at TWiM at the end of my first undergraduate summer research gig where I have had quite a bit of fun and frustration mucking about with wetland bacteria. As I've been reading about all these different bacilli, though, I've been wondering about species identification among bacteria and about what it even means to classify bacteria as belonging to different species. As far as I understand, most of the current work on species identification and the evolutionary history of bacteria relies on comparing the genomes of bacteria, looking for differences and similarities in highly conserved regions and in the presence or absence of accessory genes. My advisor told me that one shorthand for distinguishing between a strain and a species is whether or not the genomes of the two bacteria are more than 95% related to one another. This seems like a fairly arbitrary figure to me. Then, too, some species are very closely related, but lead significantly different lives in different environments. I've had the pleasure of getting to know Bacillus mycoides this summer, but I don't think I would have been able to handle this little bug if it behaved much like its more famous and dangerous relative, Bacillus anthracis. It seems to me that any species classification must take lifestyle, metabolism, behavior, environment into account. So how do all of you microbiologists think about this notion of species? How do you distinguish between a genus, a species, and a strain? Is it primarily down to differences in the genome, or do you care mostly about those differences which lead to substantially different ways of living? Thanks so much for reading, your faithful listener, Alexandra.
Very thoughtful question.
Do you know the answer?
Oh, I think you could probably get 10 different answers if you asked 10 different microbiologists right now. But I think everybody appreciates that as we understand more about bacteria and have more access to genome sequences, that it's getting harder and harder to come up with a simple definition.
So my view is that for years we cataloged bacteria by things we could see and measure, not only by morphology, gram stain, colony formation, but biochemicals. And then as we got the ability to sequence ribosomal RNA, 16S ribosomal RNA, we then could see how a lot of that made sense and a lot of it didn't. And so there was a lot of reclassification based on the 16S RNA. And when you do that, you end up saying two bacteria are related, which may have very different lifestyles. And the same thing happens in virology. The viruses I work with, poliovirus and rhinovirus, infect the intestinal tract polio or the lung rhinoviruses. They're very different, yet the sequence tells us they're very related. So basically a very small difference in sequence can cause a difference in how the microbe grows or behaves. So I think the sequence is very important, and you can now identify new organisms, sequence them, and immediately put them in the right family and genus and species based on just the sequence.
Phylogenetically, right.
Yeah, phylogenetically. So she's asking, is this the only thing we look at? And sometimes it is. I know when we get new viruses, immediately you look at the sequence, you say, oh, yes, it belongs in this family. Without even being able to culture the virus, right? We can do that.
Right. So as a practical matter, we are changing the names. Bacteria that were known by one name years ago based on their appearance or their biochemical profile. Now that we have more information, we're giving them new names,
right?
Formerly known as.
And that's also where this operational taxonomic unit comes in, right?
Right.
You assign it by sequence, and it's operational because there are a lot of things, in these microbial communities of the gut, for example, you can't culture most of the members, so you put them in units based on the sequence.
But as she points out, now the species name is not necessarily a good predictor of where that microbe will grow or how it will grow.
Absolutely. So I fought the sequence classification for a long time, but now I'm realizing that, if you think about the phylogeny and how, two viruses have a common ancestor that they evolved from, for example, the sequence makes a lot of sense.
So it'll come down to evolution rather than biology.
I think so. I think it has to. I think, things can become very different biologically with a few changes, but they're still very much related.
And then as a practical matter, the way we deal with it, for example, with E. coli, there are a number of different what we call pathotypes. So E. coli that live in the urinary tract versus extra intestinal sites,
right,
et cetera. And so we come up with different pathotypes, but they're all the same E. coli species.
Yeah. Although, we talked with James, the last TWiM we did at ICAC, James, I forget his name, James Johnson, who works on extra intestinal pathogenic E. coli. And he said less than 40% of the genome of all the E. coli are a core E. coli sequence. The rest is really different.
Yeah, that's the other issue. There's all this horizontal gene transmission, and so it's changing with time also.
Yeah. And then things are all mixed up and
right.
Who's what. It reminds me of the area code situation with phones. It used to be you moved to Michigan, you got an area code. And if you moved to New York, you got another area code. And then you could tell where people are from by their area codes. But now it will be that you can move around the country with the same area code. So you won't be able to know anymore. I don't actually know what that has to do with this, but it sounds similar. All right. The next email is from Peter, who writes, Dear TWiM team, I thought this was worth a mention. The fungus Cholara Fraxinia causes ash dieback disease and is killing many ash trees in Britain and Europe. Losses in Denmark are believed to be 60 to 90% of all ash trees. Dr. Dan McLean of the John Innes Center in Norwich came up with the idea of crowdsourcing the analysis of the fungal genome and the genomes of susceptible and resistant trees through a Facebook game. And he gives a link for that. The hope is that the detailed analysis will give clues to the origins of the disease and help identify fungus-resistant ash trees to grow in the future. Top gamers may have their names published in the scientific articles for their role in helping analyze the genetic information. Results of the project will be made available on the crowdsourcing website OpenAsh Dieback. So this is a cool way of using the crowdsourcing to solve a problem that for a computer is difficult and takes a lot of time. The human eye is very good at picking up patterns. So basically they align sequences and they want you to pick up the differences. And this is shown to be easier for people to do than computers sometimes. They're looking for open reading frames and that sort of thing. And they have a lot of genome sequences from these various trees. So isn't that cool?
It is. And I'll bet some people try to come up with algorithms on
Sure.
their own. So it's neat putting science in the hands of the people.
Yep. Our next one is from Andres, who writes, Hi, TWiM team. I love listening to all your shows. TWiV, TWIP, and TWiM on Stitcher Radio. Thank you very much for the many hours of entertainment and learning. On TWiM 61, it was mentioned that the body needs gut bacteria to make vitamin K and B. If you have not had a show on these bacteria, I would like to suggest that these would make a good show topic. Keep up the great work. I think the gut microbiome is going to be the topic of many shows. Andres.
As it should.
As it should.
And thank you, Andy.
And John writes, this is an interesting one. In TWiM 61, he talked about a species of insect with a symbiotic bacteria, which itself has a symbiont, and which also had a lot of bacterial genes that have been taken up into the insect's DNA. My understanding was that bacterial DNA and eukaryotic DNA have some differences that make it hard to move between the two. Do those differences cause a problem for movement of genes between insects and bacteria? If so, is there any indication of what has to happen for the gene transfer to work and how common that is? At the risk of slipping into TWiV territory, it seems like these endosymbionts would provide a lot of opportunity for infection of the host cell with bacteriophage? Are there examples of phage infecting eukaryotic cells? Thanks for answering my amateur questions and for your wonderful podcasts. Well, as far as phage infecting eukaryotic cells, actually, this is also a question that came up on TWiM today. I don't know of any examples of that. It's very hard for the phage to get into a eukaryotic cell, and once it gets in there, it doesn't seem to work. Although certainly components of phages work in eukaryotic cells, promoters and various polymerases, for example. We use T7 RNA polymerase in eukaryotic cells all the time.
Actually, the bacterium that I study, Legionella pneumophila, which causes Legionnaire's disease, grows inside amoeba, which are eukaryotes. And the bacterial genome is filled with sequences that are classically identified as eukaryotic DNA. So the transfer can happen both directions. So
there's no block really to transfer?
I don't think I would go that far. We don't understand what the barriers are in Legionella, but as John points out, bacteria do have defense pathways to protect their genome from random bits.
Right. Yeah, I don't know what he means by differences that make it hard to move between the two. You know what he's talking about?
My guess is he's referring to restriction modification systems, methylases, etc., that make a bacterium able to recognize its own DNA and foreign DNA, distinguish the two.
Yeah. So obviously nothing is perfect. So there's a workaround for everything. So it does happen.
And the cell might want their protective barrier to not be perfect, because if you take up a little bit of new DNA, if at some low level the population takes up new DNA, that's an opportunity to acquire a new trait that could be beneficial.
Right.
So I can see how you'd want there to be some slop in the system.
Yeah. Biology is always sloppy. That's how evolution happens, right?
Right.
So this paper that John is talking about is one that Elio discussed, where there's what do you call those bugs? Those pill bugs, the ones that roll up. And they have within them a bacteria, which is a symbiont, and that has within it another symbiont. And the three of them are able to do the metabolism that's needed. No one alone can exist. But the bugs survive on just sap, which isn't enough to provide everything. No amino acids, so the bacteria provide that. And there's a metabolic pathway that is made up of the two bacteria genomes and this bug genome. It's amazing. Just amazing. David writes, in TWiM 60 during the email segment, there was a discussion about preservation of DNA in amber. Regardless of Michael Crichton's book Jurassic Park, written in 1990, DNA preserved in amber has not panned out. Amber, which preserves in great detail the outer structure of insects, rarely preserves any internal material. Essentially, organisms in amber are casts. In addition, amber is porous to air, and air is corrosive to DNA. Searches for DNA in amber have not found much. Early 90s claims of DNA found in amber have been disputed. Vincent may recall my email answered on TWiM episode, I don't remember which one, about viruses trapped in amber. The article was behind a paywall, so I was not able to read it, but I'm pretty sure no DNA was recovered. For your information, DNA from bone has had more success. A recent announcement was made that DNA was recovered from a 700,000-year-old bone as frozen horse bone. So ancient DNA recovery is still possible, not just from amber. And he gives a link to a YouTube video of a paleontologist discussing DNA from amber. That's pretty cool.
What a great audience we have.
Isn't it great?
Yeah, it's terrific. Thank you.
You're just beginning to see how absolutely cool this is. And the next one is from Christine, who writes, Dear Vincent and fellow TWimmers, I really love your podcast. My son told me about them, and I have worked my way through the shows. I've been learning so much. I recently read that children with folic acid deficiency may have a higher incidence of regressive autism, and it got me wondering about the microbiome and nutrition. I heard Michael and maybe Joe mention that the microbiome supplies 5 to 10% of your calories, and I found review articles that include references for that, but I haven't found a good review of micronutrients the microbiome supplies, like the B vitamins. Would you consider having a TWiM on the microbiome and the nutrition it supplies? I think we just had a request.
I think we did.
All right. Since children with autism often have problematic microbiomes, could it be that they aren't getting the right vitamins at the right times for their brain development? Can the body ask the microbiome for more vitamins when it needs them through signaling? Could the microbiome supply much larger amounts of vitamins than we realize? Perhaps there are micronutrients that we don't know of yet because our microbiota supply them instead of our food. I'm interested in the vitamins because of my own story. You could leave this part out if you want to, but I think it's interesting. I used to be a bioengineer working in microscopy, gene arrays, and bioinformatics, but I suddenly got sick with a disabling mitochondrial disease in my mid-40s. I could run a mile without thinking about it. Then a year later, I couldn't sit up for 30 minutes. Then a year after that, I was going into heart failure. I started taking very large amounts of riboflavin, and my heart, which had become enlarged, shrunk back to normal size and started working just fine. Unfortunately, my other muscles didn't respond so well, but I'm still alive 5 years later, which wouldn't be the case without the riboflavin. Some of my friends on my mito and FOD fatty oxidation disorder support groups also helped a lot by vitamins, and it makes me wonder, why did it change? I was doing fine for 45 years, then suddenly I need a bunch of riboflavin to survive. I'm going to suspect the microbiome. I like how you think about it as another organ. Thanks for your great work, Chris. She gives links to review articles on the microbiome, obesity, which you heard today, folate deficiency. P.S. I love Elio's voice. Sorry he's not here. And PSS, my genetic defects are normally mild ones in nuclear genes affecting fatty acid oxidation and complex 1 of the respiratory chain, so the odd inheritance and aging effects on the mitochondrial DNA disorders don't really apply to me. Interesting. Well, I think she's on to really possible things that we just don't know about, right?
Yeah, and I think these are questions that the field is interested in tackling, better understanding what the microbiome contributes.
Right. One more here from Joe. Dear TWiM folks, I'm delighted that you are finally devoting the time and attention to the most important microbiology of all, yeast fermentations. Your letter last week from Mark on yeast inoculations reflects a very modern North American perspective on winemaking. Inoculations with clonal yeast strains are, of course, quite modern. Wine was made for millennia without inoculation. Many of the finest wines of France, Vincent, are fermented with their indigenous yeasts. A complex microbiome produces a more complex metabolome, unsurprisingly, giving wines of distinct complexity and subtlety of flavor. One man's complexity may, of course, be another's excess funk in some cases. Taste is always paramount. It's also true that choices in the vineyard may influence the success of native ferments. Heavy spraying with fungicides, for instance, surely selects yeast populations, but so does weather during the harvest, tying the wine more closely to its vintage. The requirement of highly alcohol-tolerant yeast to finish the fermentations of high-sugar grapes reflects some climate change, but it also reflects a recent cultural preference in California for riper and, to my taste, simpler fruit flavors that come with grapes left on the vine later in the summer to eliminate green flavors and coincidentally raise sugar contents. I hope I have the chance to wave from the audience at a live TWiM or TWiV this week, and I hope you will continue to give alcoholic fermentation the attention they surely deserve. So Joe was at ICAC in Denver.
And did you wave? Did he wave?
Didn't see anyone wave, but hopefully he was there. We've had an interesting little discussion in the last few TWiMs about winemaking.
There's a lot of interest among our readership, that's clear.
And we started with a paper we did, and Michael saying that, wines are made by inoculating them with yeast cultures. And then we had someone write back saying, it's not always. It's been done for many, many years without inoculating. They use the yeast that grow up on the grapes on the vine. And here he's saying the same. They're fermented with indigenous yeasts. Now, someday we need to do an entire episode with an expert on winemaking. Although I suspect there'll be many different.
We should do that when we're together, and not just from our offices.
So we can sample.
I think so.
Now that's a great idea. In fact, this letter is making me thirsty, so I think it's time for us to wrap this up, so we can get back to work.
All right.
This episode of TWiM will be found at microbeworld.org/TWiM as usual, and also on iTunes. And if you like what we do, go over to iTunes and subscribe and leave a comment or a rating there in the directory. It helps to keep us visible. And we like that so that more people can find out about what we do. And do send your questions and comments to us. You can email them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thanks for joining me today, Michele.
My pleasure.
Do you enjoy doing TWiM?
I do.
And it gets better.
And I'm learning a lot. I really am. It's great. But I'm also happy to hear some feedback from our listeners.
Oh, I'm sure it will be just wonderful. But yeah, stick with us and you'll see our wonderful listeners, and I'm sure they will start to make comments. So if anyone has an opinion about Michele, let us know.
Until next time.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and Chris Condian and Ray Ortega for all their technical help. The music that you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. We use it with his permission, and you should go to his website, ronaldjenkins.com, and check out all the cool stuff that he does. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.